We are really happy to have Sabine Kastner today. She is a professor of neuroscience and psychology at Princeton, and she has really unique backgrounds in that she has background in both human neuroimaging and animal neuropsychology and neurophysiology. So she's done postdocs in both of those fields, and her work is actually integrating those fields, which is really unique and really needed, in my opinion. Her work is mostly focused on large-scale networks, especially when it comes to visual attention and visual processing. And we're going to hear a lot about that right now. So give a hand to Sabine.
Thanks very much for having me. I'm really excited to spending the day with you today and to share some of the data that we have collected during the last several years on the electrophysiology of the primate attention network. So let me just introduce you to the issues that we study in the attention field by using this painting here by René Magritte, who is one of my favorite artists. So I hope you can sort of see that. If you would lower the lights in the front, it would be more visible. What you look at here is the canvas that the painter left in front of a window. And we make a lot of assumptions about what is behind that canvas. And probably most of us would think that it's exactly the scene that the artist has worked on throughout the day. But there's, of course, very little that warrants that assumption. And what Magritte captures here, and in a lot of his other work, is really how vision works, that it's a back and forth of assumptions that we make about the world and what is really there. But I think at the end of the day, we draw on very few clues from the physical world and fill in a lot of the important rest. And this is a very neat dimension of our field of neuroscience. In general, it gives us a philosophical perspective. And Magritte knew about that philosophical perspective on vision very well when he called this piece the human condition. Now, let me now switch to the attention field and the problems that we have in the attention field by just starting from this scene here. So let's treat that just as a complex scene, this kind of scenes that we have in our visual environments. Now, the way we take information from complex natural scenes in and how we select information from scenes is very often portrayed by something like that. We use what people call in psychology textbooks a spotlight of attention. This is a spatial mechanism. It has some kind of extent, and we can move it dynamically around to different parts of that scene. In real time, this goes much faster than in my animation here, and eventually we gather the kind of information that we need to reconstruct the scene. And this kind of spatial mechanism, or the set of spatial mechanisms that underlies this kind of selection, is the topic of my talk today. Now let's take a look first of all to see what kind of network is engaged. And this is in the human brain. This is from an imaging study during a spatial selection of the sort that I've just demonstrated for you. Let me see if I get this movie to work here. So I explain it while it runs. So this is a right hemisphere. This is the posterior pole. This is the frontal pole that we reconstructed here. And my movie has a problem running. Let me just see if I can do it again. So we inflated this brain surface so that we open up the sulci and can look into them. So, and what we see here is the buildup of activity. These are fMRI signals in occipital cortex. Again, the movie just takes up here, but it's not really important. So this is the visual system proper, if you like. And then a little bit delayed in time, there is this buildup of activity in higher order cortex here along the interparietal sulcus. And there are also regions in frontal cortex that would be activated that you cannot appreciate in that view. And what has fascinated me for several years now is that this large-scale network that encompasses so many nodes that you just see here at the cortical level is set up in a very efficient way. So these kind of spatial selections we can make within 200 or 300 milliseconds. So this large-scale network, this attention network, needs to cooperate in very efficient ways to accomplish its function. And we are interested how this kind of communication across the network works. Now, we study these issues in two different brain models, in the human brain that you see here on the left and the macaque monkey brain that you see on the right. And we use a variety of methods from neuroimaging and from intracranial physiology that we combine. And we do that in both species. So we do also intracranial recordings in epilepsy patients, electrocorticography, mainly together with Bob Knight at UC Berkeley and Joseph Pavizzi at Stanford. But I will focus today in my talk mainly on simultaneous multi-site recordings that we do in the monkey brain. But keep in mind that all the experiments that we do in non-human primates, we do in parallel with intracranial physiology also in humans to gather over time data that help us to directly compare these two brain models. Now, the way we typically conceptualize our spatial selections that we make is in a very simplified notion like this that you will find in many textbooks. The idea here is that information is flowing through the visual system, so it arrives here at the level of primary visual cortex and is then represented, the incoming visual information, along two major pathways lead into the parietal cortex and the inferior temporal cortex. And these multiple visual representations are then modified, so the ongoing visual processing is modulated by signals that arise in parietal cortex and in frontal cortex and are fed back. This is what we typically call top-down attention or something like that, and basically modify this ongoing visual processing. And there has been a lot of work in the field trying to understand what these modulations are. But this is a kind of our canonical model of a top-down process. And we can use that for attention, but we can also use this model for a lot of other cognitive processes. Now, what's the evidence for this kind of top-down control of spatial attention? In very early imaging experiments that we did in the late 90s, we did a very simple experiment to gather some support for this top-down model. So we did the experiment where our subjects were maintaining fixation, so they gaze at this fixation point over here, and this is just shown here schematically. They would direct attention to that location in space in what we call a covert shift of attention, so without moving the eyes. The eyes are always fixed over here. And later on in all the attention tasks that I will use in this talk, this is always a covert attention task, so it has the same principle. Now, we instructed our subjects to direct attention to this location in space and then to expect the onset of a visual array that's shown over here and then to attend to that location and make a visual discrimination. And what we found in this fMRI experiment in humans was that in higher order cortex, here are data from the frontal eye fields, there is a very large increase in activity. Once you allocate attention at that location in space, there is no visual input yet, which is then sustained when you actually make visual selections during the period when there is visual input. So this kind of sustained activity mainly reflects the cognitive operation here, the allocation of spatial attention, rather than the processing of visual information. Now, in lower order cortex, this is an area here at the intermediate processing levels in visual cortex, area V4, you can see that there's also this increase during this expectation period when attention is allocated, but then there is a boost of signals once the visual stimuli are actually kicking in. So here you get this kind of two-stage profile that these visual areas show, and this way you can basically make out a top layer that reflects the attentional operations in frontal and parietal cortex, and then this visual processing layer that seems to be more engaged in visual processing. And that was one of the data points that we had for assuming such a top-down model of attentional control. Now, there are much more conclusive data from animal electrophysiology. This is a monkey physiology study from Tyrone Moore's lab, but I will not put you, in the interest of time, through all of this, otherwise you don't get enough of the new data that I want to show you, but just to let you know that this is really a very, very well-established concept with lots of interesting and conclusive data. Okay, why do I tell you all of that? Simply because I want to challenge that concept today in my talk, and I want to tell you in the first part that we think that there's not only top-down control that comes from frontoparietal cortex, but that there's a lot of control from the center. The center of the brain is literally the center, that's the thalamus, and I will tell you a little bit about that. So that will be the first part of my talk. And then in the second part of my talk, I would like to challenge the second notion in attention research that I introduced with a Magritte painting in the beginning, and that is the one of the spotlight of attention. And I will show you actually that this spotlight is really an overcome concept and that we need to redefine or reconceptualize it. We'll show you that there are actually multiple dynamic spotlights that we use when we make a very simple spatial allocation of our attentional resources. Okay, let me start with the first one, the first part, the control from the center. By the way, if you have a question, any question of any sort, I know that you're a very diverse group here, a very interdisciplinary group. Just speak up and ask the question right away, okay? So I will talk about the role of thalamo-cortical interactions in visual attention. This is a schematic here of the thalamus. This is a group of nuclei. And in the visual domain that I will talk about today, there are two nuclei that are particularly important. And one you may have heard about, that's the lateral geniculate nucleus over here. This is the relay nucleus that transmits the information that comes from the retina to mainly primary visual cortex. And then there is a second nucleus, that is this one over here, a very large one, the pulvinar nucleus, and that nucleus is the largest thalamic nucleus in the primate thalamus. And it undergoes a tremendous growth during evolution on the order of what prefrontal cortex undergoes. The pulvinar, unlike the lateral geniculate nucleus, receives a significant amount of input from the cortex. It receives very little input from the periphery. So it interpatches the thalamus and the cortical networks. But we do not know much about its functional roles. Now, when we look at that kind of connectivity that the pulvinar has with the cortex a little bit more closely, there's actually something very interesting. So here I show you two visual areas, V4 and TO. This is the 6 layers of the cortical sheet. And what we see here is that these two areas share a direct cortical-cortical connection that is indicated by this arrow here, which goes from the superficial layers to the input layer 4 in the subsequent area. Now, the interesting anatomical rule, and that holds for any two areas that have this direct cortical-cortical connectivity in the visual system, is that you have this connectivity, then you have also an indirect loop that goes through the pulvinar. So the pulvinar, in that way, is basically made up of little patchy projection zones. Here again, these in red, area V4, and black area TO, and you see that they intermingle here. And these are zones in the pulvinar where these two regions can talk to each other, but we know very little about the functional role of this kind of intricate connectivity that we find in the connections of cortex and pulvinar. And we wanted to understand that a little bit better in the framework of visual attention. And this is work that Yuri Salmon and Liang Wang have done in my laboratory. Now, what we wanted to do is we wanted to train a macaque monkey in an attention task and then record simultaneously from two interconnected cortical areas. So we chose actually those two areas before in TO, which are kind of mid-level areas along the visual processing hierarchy, and then to record from the projection zone in the pulvinar that they share. And we had to do a lot of imaging to actually visualize this projection zone in an in vivo preparation. But I will not tell you about it. If you're interested, I can discuss that later. Now, in these studies, we tested a very specific hypotheses, and I want to introduce you to some of the background knowledge that we drew upon to formulate the hypotheses. So one piece of evidence that we drew from is that spiking activity, which is indicated here by these vertical bars, is very often associated in any cortical region with certain phases of an ongoing oscillation. This is depicted here. So here spiking activity would occur more frequently with the peaks of that oscillation and not the troughs. So the idea is that these oscillatory processes open up windows of relative excitability and inexcitability for the local populations in these areas. Now, if an area 1 here wants to propagate spiking activity to area 2, it is kind of convenient to bring this oscillatory pattern of activity into synchrony so that a spike that propagates from here to here can meet a relative excitable window and not an inexcitable window. So we would call this an effective phase relation for communication. And this is opposed to this kind of scenario where they are out of sync and where you have a relative ineffective phase relationship for communication. So that is one concept that we drew from. So the problem here is how can the synchrony between these two regions that want to communicate with each other, how can that be set up? So there is one notion, and that's heavily discussed at the moment in the literature, that talks about feed-forward and feedback pathways that can be used in order to kind of negotiate the best synchrony and the best temporal pattern for these two regions to communicate. But then there is, of course, a different alternative hypothesis here that we were testing, and that is that there could be a third area that is connected to both of these that could set up this kind of synchrony between these two regions. And this is the hypothesis we are probing here for the pulvinar. So the pulvinar may synchronize neural activity in two interconnected cortical areas in order to optimize information transmission between them. Now let me introduce you to the task that we used to probe these issues. We drew here from a task that is widely used in human cognitive psychology. It's the Erickson flanker task. And we did that again because we do all of these studies in parallel with humans. And we hope also that we can bring the macaque primate model closer to the human brain that way, rather than using just some arbitrary task. So the task goes like this. The animal catches fixation here centrally on this fixation point. And then there is a location marker that is shown in one of 6 to 8 locations that are arranged in a circular manner around fixation. It's followed then by a variable delay period up to a second roughly, during which the animal holds this spatial information online. And this is along those time scales typically done by simply sustaining attention at that location. And then there's an arrow composed of 2 shapes, barrel and bow ties, and the animal indicates with a manual response whether there's a barrel or a bow tie in the cued location. Now, the reason why this is called a flanker task is that the target stimulus can be flanked by either dissimilar stimuli, like in this example, or by identical stimuli. And depending on these flanking conditions, there's actually different outcome behaviorally. This is here for a human observers first. You can see here that in this congruent case, so identical flanking targets, there is a reaction time benefit relative to the incongruent condition. This is reaction time. And this overall effect also depends on the number of distractors in the display. So this is very classical human behavior that has been shown in hundreds and hundreds of studies and variants of the flanker task. Now, the monkeys show this flanker effect as well. And that was obviously one of the first things we wanted to establish here. So you see this exact same reaction time cost or benefit depending on the flanking conditions. Now, let's look first what the pulvinar, that is the structure that we know so little about, is doing in a task like that, because it may not be involved at all, obviously. Yes?
Reaction time, is there also a corresponding relationship with the errors that are made as well? They make errors.
They make errors. They make also more errors as well. Yes, that goes together. So let's look what the pulvinar is doing just at the level of single units here. So that's the first thing that I'll show you. But keep in mind, we are doing the simultaneous recordings from these two cortical areas as well. But let's just look at the single unit activity first. The way we look at these data, and all the analysis that I'm going to show you after this will have the same format, is that we are looking at a receptor field that's outlined over here in red that we are recording from under the conditions when we present the cue here, and the animal attends to it and does the attention task in this location, versus when we cue this blue field here and the animal is performing it on the opposite field location. We're still recording here, but then under conditions when the animal directs attention elsewhere. So that will be our control condition denoted here in blue. Now, we can look at these different conditions here. So the cue evoked activity is shown here. So it's a very nice cue evoked activity, which you would expect in a visual structure, which is followed then by elevated delay activity. So this is the period here when the animal just sustains attention at that remembered location. So this is not contaminated by visual input at this moment in time. And this is actually a period that I will focus on later on when I show you some kind of interactions that are built up between these areas during that period. And then you see when the target area is presented, a moderate increase in neuroactivity here when the animal attends to that receptor field that we are recording from relative to when it attends elsewhere. So this kind of profile is a very classical profile that we have seen in similar tasks over and over at the cortical level. And that was a nice sanity check here that this thalamic structure is also part of that attention network and has some role in processing information here. Now let's look at the simultaneous recordings now and at the network interactions between these three hubs of the network we're looking at here. Now, when we're looking at network interactions across areas, we look at local field potential, so at the activity that is produced by local populations. This is how a local field potential may look like in the raw form. So it is composed of many, many different components, and you can break these components up in frequency space that is shown over here. This is the time, and then you get components of different frequency bands, ranging, from the alpha band in the visual system, an important one between 7 to 12 hertz, all the way to faster components like the gamma band or even high gamma activity. Now let's look at the pairwise comparisons between these two regions at the level of local field potential, and let's see whether attention will modulate the synchrony between neuropopulations in these areas. And we first looked at the cortical level, the two cortical regions. It has been shown that attention modulates synchrony patterns in numerous tasks. So we just, again, wanted to run here more of a sanity check. And indeed, we find, and this is all during the delay period, so that's during the period when the animal sustains attention, there's no visual input, there's no contamination also by onsets of signals and so on. And we see here that there's an increase in synchrony in a frequency band around 10 hertz between the two cortical regions, V4 and TEO, when the animal attends to the receptive field. Again, in blue is attend away. Now, how does that look like, these kinds of interactions between the cortex and the pulvinar? Now, what we see here is that there's a very similar pattern. There are these increases in synchrony around 10 hertz between the pulvinar and V4, and also the pulvinar and TEO. So what this basically tells us is that these three regions here, these three areas, are part of the attention network, and they seem to use a 10 hertz frequency band to communicate during this period when the animal maintains that spatial information and sustains attention. So this circuit here somehow uses that alpha band. But now the interesting question is, what is the direction of information flow in this mini network here? Is this information that flows from the cortex to the thalamus or vice versa? And we wanted to approximate that by using a statistical model to look at this issue. We probed here Granger causality. We have done in the meantime also inactivation studies, activating the pulvinar and looking at what the cortex is doing. And these two sets of studies, the other ones are unpublished and also not quite finished yet, so they are preliminary. But these two pretty much converge on the same message. So the Granger analyses, I think, tell us the right hypotheses here. Now, when we look at the Granger causal influences that the pulvinar has on the cortical regions, what we see is that there is, again, a stronger influence during attention in this 10 hertz band that arises from the pulvinar and imposes on the cortex, and that is also true for the Granger causal influences of the pulvinar onto TEO. So this is basically this indirect pathway that could be used to set up this kind of communication between the pulvinar and the cortex. Now, the cortex also has direct pathways, as I've shown you in the beginning. So it has feed-forward and feedback connectivity between these two regions. And so one question for us was, what are these direct cortical pathways are doing in this task period when the attention network is basically set up for functioning? And as you can see here, these are the Granger causal influences of V4 into TEO and vice versa. The cortex does not really use, or does not appear to use, these direct cortical pathways, but most of the information during that period of time seems to go through the thalamus. Now, what does this all mean? So I have shown you that the pulvinar may be a structure that can synchronize oscillatory activity in cortical networks. And we have seen that at the level of visual cortex. We have a data set that also shows the same thing in an interaction between a higher order area in the posterior parietal cortex and a visual area where the pulvinar does basically the same thing. So it can basically organize the temporal activity in cortex, or the temporal structure that cortex will use for subsequent processing. And this seems to happen, at least in this task, and this may be task dependent, in this 10 hertz frequency band. So what we think what we need to do in our models of attention is that, in addition to probing the idea of top-down control of a frontoparietal network that is certainly existing, so I'm not contesting that, we need to think about other control mechanisms that are used during selective attention. And one is this temporal control function where the cortex and the cortical networks get structured in time. And this is the kind of function that for now we associate with a pulvinar, and this is why I call it here a timekeeper. So this is what I wanted to tell you about thalamo-cortical interactions and the control functions that they may have in selective attention. Yes?
Do you think that the role of the linear in attention is same if the view that you're presenting was from a different modality? Let's say you have a film that indicates the moment to focus on the left side of the screen versus the right side. So do you think this is due to the fact that the cue is a visual one and that when you present the cue or react to the network, is it that we reacted with the network?
That's a very interesting question. So the pulvinar is mainly visual, but it has, particularly in the dorsal parts, some multimodal neurons and parts. So it could be that, but of course we don't do that experiment. I don't think it would matter, actually, in the end. I think that those parts could be recruited to take care of that issue and to basically reset the network in the same way. But it would certainly be a very, very interesting experiment to do. Are there other questions? Yeah.
Do you see a lot of trial-to-trial variability in the spectral content of those different areas? And if so, do they track one another on a single trial basis?
That's a great question. So that's one of the issues, I think, that we are really, really grappling with in the non-human primate field, that basically single trial analyses are way too noisy typically to do these sort of analyses on. So these are all group analyses that I've shown you that were averaged over many, many trials. We have tried a single trial analysis. And that's very mixed. It really depends on the day. And in some good sessions, you see robust single trial patterns, but it's really something that is very difficult to do in monkeys. So I wouldn't put too much stock into single trial analysis at this moment in time. So the human ECOG data are actually more conclusive on those ends, but there we do not have the deep recordings typically. Other questions? Okay, then let me continue with the second part of my talk here. So I have shown you basically in the beginning that there is a spotlight of attention, but that this spotlight is basically a stationary one. You can move it around, but once it's allocated, it is stationary and continuous. And what I want to show you, if I get there in my slides here, is that it really works quite differently. So once we allocate attention somewhere, what happens actually are two things. One is that the spotlight starts to blink. So it goes through phases of increased or decreased perceptual sensitivity. So it's not a stationary spotlight, it's a dynamic spotlight. But the second thing that happens is that involuntarily, so that's something that is automatized and we do not control, we open at least one more spatial mechanism that can monitor from other locations in the field. So here are two outlined, but there may be 3 or 4 that a second spatial mechanism can monitor in a rhythmic way, like shown here. So the spotlight is dynamic, and it's not a single spotlight, but there are multiple spotlights when we allocate attention just here. So that's the message that I want to get across. So if you lose me, just take that take-home message. But I want to now show you how we have studied that in the lab at the experimental level. So this started when we wanted to establish a neural basis for object-based attention in non-human primates. And the task that we chose is, again, a task that is known very well to human cognitive psychologists. It's the Ugly driver task. That's a very simple task. You get 2 objects. There's a central fixation point, and you always keep your gaze here, so it's, again, a covert attention shift. And then there is a cue that's presented in one of these 4 corners here on one of the 2 objects, which is followed by a threshold detection task. So there's a variable delay period, and then there's, I put this here really super threshold so that you can see it, but it's a very subtle change, and that occurs with about 75 or 80% validity in the cued location. So the cued location is heavily biased in this task. But sometimes this detection or this cue presentation can happen either on the same object and on the opposite end, so that would be in this constellation down here, or it can happen on the second object that would be here, and that would be equidistant to the cue. The presentation of these two objects could also be in this horizontal fashion. So this is just one example, and it all changes from trial to trial. So what you can do in this task is that you can now track behavior at these three locations. And again, this is just one constellation. The cued location, then the infrequent same object location, and the different object location. And these occur at the same frequency. So they're infrequent, but there's no bias between one or the other. And now what Driver and Agley have shown in their task was that there's actually a bias towards the same object. So you're better to make detections at the same object than at the different object. And that's called an object-based attention effect. And the idea is that once you allocate attention at that object, remember the cue was up here, then attention spreads within the boundaries of that object. And this is why you get an advantage at this location over this location. Now, when we started to look into this task, we looked into the literature, and we saw that sometimes this effect is found quite clearly, and other times it's not. And the only so this basic task is always used, and the only thing that seemed to be different from study to study was the length of the cue target interval. So that's the interval between the cue presentation and the target presentation when there is no change on the screen and only the objects are there. In all studies, there was only one particular interval used, but these intervals were different. There were 300, 500, 700 milliseconds, and so on and so forth. So we wanted to learn whether there was something going on with that cue target interval, and we wanted to probe that in a behavioral study. And that's what we did in humans. And I show you the data here for 14 observers. This is work that Ian Fiebelkorn has done. And these are just the raw data. This is the detection proportion as a function of the cue target interval that we binned in very, very small bins here. And in black, I show you the cued location, in orange, the same object location, and in blue, the different object location. And you can see here several things. So there is a very clear difference. For instance, at the cue target interval here, around 450 milliseconds, you're particularly good at the same object, but not at the different object. And then that reverses, and around 550 milliseconds or so, actually you wouldn't get an object-based effect, because now this object-based effect seems to be somewhat covered here. So there's a very clear oscillation that seems to swing back and forth between these two locations, and you can detrend the data and make that more clear. And by doing that, you can actually see two oscillations. One is at the cued location, so I just enhance it here. Again, this is just the success rate here as a function of the cue target interval. And you see that there are also these little bumps here in form of an 8 Hertz oscillation, where you sometimes are a little bit better or worse to make these target detections. And then you see this kind of inner leaf pattern here of the behavioral oscillation that goes at 4 Hertz between the same object and the different object locations. And you can quantify this further by simply subtracting those, and you see very clearly these troughs and peaks here of that oscillation. Now, we have now two rhythmic processes here in this attention task. One is what we refer to as rhythmic sampling at the attended location, and the other one, what I call rhythmic monitoring outside the focus of attention, that is going back and forth between these two locations. And this is an automatic process. You're not aware of this process. This is just happening automatically. So at that point, I thought, after we had all these human behavioral studies done, that that was the end of our monkey experiment, because I could not possibly envision that a monkey would show this type of behavior. But as it goes, sometimes you're wrong, and here are now the results from two animals, from two monkeys. This is the rhythmic monitoring outside the focus of attention. And they show the exact same thing. Even the time windows are exactly aligned. And this made actually this kind of experiment a lot more interesting, because what we, in the first place, learned from that is that these rhythmic properties of selective attention that we tap into here in this task are preserved throughout evolution. And that makes them a lot more foundational. Now, let me just summarize that really briefly before I show you just very, very few preliminary results on the electrophysiological recordings in this task that we have done in macaque monkeys. So I've shown you that visual spatial attention is a highly dynamic process. I don't think this is restricted just to this task. I think you will find that in any attention task, in any spatial attention task at least, if you look for it. So we see that there are at least 2 concurrent spatial mechanisms, and we see that there are several rhythmic properties. And these rhythmic properties certainly depend on the task demand, so I think they can be very flexible. And the rhythmic properties of attention are preserved throughout evolution. And this is just a side note for those psychologists of you in the room. We know a few of these phenomena in the attention literature that we refer to as inhibition of return or the attentional blink. And I think they just tap into these rhythmic properties, and there's nothing else that underlies them. But this is just, a blank statement, and I hope that somebody will be there one day to fill it with life and do the experiments that need to be done to show it. So let me just show you some very preliminary results that we did in 3-way recordings between 2 higher order regions in the macaque brain, the frontal eye fields and frontal cortex, and the lateral interparietal area in the posterior parietal cortex. And we also recorded from the dorsal pulvinar that these 2 regions are interconnected with. So I show you just single cell data first, just to show you that these regions are actually engaged during this task. These are all regions except for the pulvinar that before had been established in spatial attention tasks, so this is not a surprise. We see here the cue onset. These data are normalized, so 0 baseline would mean no effect, so this is elevated delay activity during that period of time between the cue offset and the target onset, and this is the target onset. So that's the posterior parietal cortex region. In comparison, the pulvinar shows almost identical patterns here, and then the frontal eye fields that also show very similar activity. Elevated delay activity is much, much higher. So this is just a sanity check that these regions are engaged during this task. Now, as a very first step in this analysis, what we wanted to do was that we wanted to link the behavioral oscillations that we have seen in this task to neural oscillations that we can measure in this network and in these different regions. And so we thought that one way to think about the behavioral oscillations is that there is a neuronal oscillation that sets up low and high excitability. A low excitability state would be denoted by a trough, and a high one by, by a peak, and a high one by a trough, but it's completely arbitrary. And the idea is that when a stimulus is shown here at a peak, then there may be a lower likelihood of detection, whereas when it is shown, when we have this neuronal oscillation here at a trough, then you may have a higher likelihood of detection. So we had this hypothesis that nodes of the attention network that drive behavioral outcome will show a consistent relationship between the phase of a neural oscillation and the rate of the behavioral detection performance. So we looked at the local field potentials, did a wavelet analysis. I will not go into the details here for these kind of phase detection relationships. So, sorry, but I want to show you this. So this is the phase here of some kind of oscillation at some frequency, and this is the hit rate, the success rate. So if there is a constant phase detection relationship, then you would think that there's a profile like that, where you get a peak and a trough. Whereas if there's none, then you get something that's flat and not related. So that's kind of what we're looking for here. So I show you data first from the pulvinar. We are looking here at a neuronal oscillation at 8 Hertz, and we look for this kind of phase detection relationship, and you see a very, very nice profile with a peak that is flanked by these troughs in the pulvinar, and this is at the cute location. Remember, the sampling at the cute location happens at 8 Hertz, and here we see that there's a relationship to a neuronal oscillation of that behavior. Now, when we look at all 3 regions here, and we have looked, of course, across the entire frequency spectrum, and there are also other frequencies that play a role here. I just kind of hone in on this one at the moment here. Then we see that we get this kind of profile also in the posterior parietal cortex and the lateral interparietal area, but we do not get it into the frontal eye fields. So what we think is going on here is that the sampling at the cute location is a result of some kind of interaction of posterior parietal cortex and the dorsal pulvinar. So what about the second process, the rhythmic monitoring outside the focus of attention that I mentioned in the behavioral data? Now here we are looking for this kind of interleaved relationship between the detections that can be made at the same object and the different object location. And here we are looking for an interleaved relationship at the level of a neuronal oscillation. In orange, you see the same object location. In blue, the different object location. Black here as a reference is the acute location. And we are looking at a 4 Hertz neuronal oscillation. And again, we get this interleaved phase detection relationship. That's a success rate that we were looking for. And we see that in the frontal eye fields. And we see that we do not see that in the posterior parietal cortex. So there you get a pretty flat profile. But we also see that in the pulvinar, this kind of relationship, and it is phase shifted, which has certainly or most likely some meaning, but we haven't looked closely into that yet. Now, what that tells us is that the rhythmic monitoring outside the focus of attention is some kind of interaction that happens between these 2 structures, the frontal eye fields and the dorsal pulvinar. Now, at first glance, that was a very surprising result for us because we thought that the allocation of attention at the acute location would be something that frontal cortex probably is doing and not posterior parietal cortex, and that this automatic process, the rhythmic monitoring, should be something that probably posterior parietal cortex is doing and not frontal cortex. But if you think about what this monitoring outside the focus of attention may be, that's probably the process that's the one that gives you the behavioral flexibility. What that process is doing is basically, while you're allocating attention here, it helps search the environment for things that may happen where you actually have to switch your focus of attention that you have here to. So that is really the process that may give you a lot of behavioral flexibility, and this is probably why it is tied to the frontal cortex. So we are right now obviously looking very deeply into these complex data that we have collected to really understand what these interactions are, and we will also expand that to other parts of that network in the future to understand these rhythmic properties of attention and the neural basis better. So to conclude, I've shown you today that, and I hope I have convinced you of that selective attention is a highly dynamic process with several rhythmic properties in the second part of my talk. And we have seen that the behavioral oscillations that we observe in the Agley driver task can be linked to a specific frontoparietal thalamic network. And this is, I think, for the first time that actually a behavior has been linked so clearly to neuronal oscillations. As you know, many neuroscientists are very skeptical about them, included myself, actually. But I think here we have this opportunity to look at behavior or complex behavior that we can relate to oscillatory activity in the brain in very compelling ways. Now, to broader points, I think that our concept of top-down control is very limited, and I think we need to expand that, and we really need to think about the kind of control processes there are in the kind of in the cognitive networks that we are studying, and then try to identify neural correlates and neural network parts for them. And the second broader conclusion that I hope I could convince you of today is that I think our concept of this static attentional spotlight that we have in our textbooks is just entirely wrong, and we need to consider these rhythmic properties that I think are present in any, at least spatial attention task. We do not know whether that will also apply to other domains of attention, like feature-based attention. And we need to consider that not only into our theory, but into our experimentation, because I think our spatial selection tasks are very often very limited in the way we design them at the moment. And with that, I thank you for your long attention span.
In the task where you have two objects, there's an intended location, then there's two unattended locations, and you see all of these oscillations. Do you think you'd see similar oscillations in only the attended location if there's only one location where the stimulus could occur? Because right now, the molecule, the human, attends to the attended location, but they also need to monitor at least somewhat the other locations.
Yeah, it's a great question, and we will never find an answer to that because it's kind of difficult to probe this. Even though I think once we have neural correlates for that, we could probe it, because then we can use the neural responses to tell us what the animal is doing behaviorally, even if that behavior is not expressed, basically. So if you want me to speculate on that, I think we always monitor. And yes, our monitoring will depend a little bit on how much weight we give to these different locations. So we will, adjust it flexibly. But I think even if we are told there's nothing going on in other locations, if it's a complex enough environment that we are in, we will always monitor, because there could always something interesting happening, right? I mean, if I, of course, just use one stimulus in a Posner task or something like that, and I deprive my subjects of that complexity, I may not find that. But I think in realistic life and natural conditions, we probably always do that monitoring, and this is why it's a very useful thing to have. It gives us this flexibility to really shift attention from where we have it to something new very quickly.
So I think you alluded to this in your talk already, but it seems like what you're saying is that perhaps this secondary spotlight, it could be predictive of future eye movements or attention and so on. So is that your prediction that if you were free viewing an actual scene and captured the viewer's attention, that these other areas of that oscillate and are coherent with the primary center of attention would be predictive of where the attention is going in the future?
Potentially, though, I think the most surprising thing, or one of the more surprising things in these findings, is that you do not only open up a second spatial mechanism, but then you spread that across, at least in this task, across 2 locations, and there may be 3 or 4 locations that you may be able to monitor in this kind of rhythmic fashion. So it's somewhat predictive, but I think if you monitor 3 or 4 things, then that will be, or you have to take that into consideration, basically. So yeah, yes and no, basically. But it's a very, very complex second mechanism that we are looking at, and we do not understand it fully at the behavioral level. I think, even though people have done some work on it in the human field during the last few years, we still need to really understand a lot about what's going on.
It sounds like there may be a question from the Emory side. Does someone over there have a question?
Yes, that's true. Can you hear me?
Yes.
Yeah, it's Dieter Jäger. So I have a question about the y-axis on these behavioral effects. I saw 0.03 difference in the proportion of events detected. I wasn't quite sure what that meant.
So that is a difference in success rate, right? So these are relatively small differences that you see there in the success to detect stimuli, but you see that it has some kind of rhythmic structure, and you can link it to phases in the brain, so to a neuronal oscillation.
So, but it's generally speaking a small effect. So you're not like inattentive during one part of the.
No, no. So the effect between the same and the different objects, that is a very, very large effect size. But the one at the, so the rhythmic sampling within that spotlight of attention that you sustain, that is a relatively small effect, at least in that setting. And we don't even know whether in natural viewing conditions where you have suprathreshold stimuli, it would play much of a role. So that's another, really interesting question to explore. But there is this rhythmic structure, whatever it does, and whenever it is recruited.
Like with that, maybe if there are remaining questions, we can take them offline. Let's thank our speaker once again.